Hi, everyone. This is your People Behind the Science podcast host, Dr. Marie McNeely, and I am excited to have you here with me for episode 823 with our guest, Dr. Ganesh Anand. Listeners, if you want to get connected with us to hear about even more amazing people doing great science, you can follow us on Blue Sky at PBT Science, and I'm at PhD Marie. We have another fantastic conversation on the way from today's guest, so I hope you're ready to meet another one of our remarkable people behind the science.
Every day, discoveries are made that will change our understanding of the world around us. Dr. Marie McNeely is here to bring you the brilliant minds who are making these discoveries so they can share their incredible stories and take you on an amazing journey. Welcome to People Behind the Science.
Hello, everyone, and welcome to People Behind the Science. Today, I am excited to speak with our guest scientist, Dr. Ganesh Anand. So, Ganesh, welcome to our show today. How are you?
I'm great. Thank you for inviting me.
Well, Ganesh, we are thrilled to have you with us today, and I'm excited to learn more about you and the work that you've been doing. But before we dive into those details, let me first take a moment to tell our listeners about your current positions and the steps that you took to get to where you are today. So listeners, Ganesh is an associate professor of chemistry, as well as biochemistry and molecular biology at Pennsylvania State University. He's also an elected board member of the International Society for Hydrogen Deuterium Exchange. He received his bachelor's degree in pharmacy and his master's degree in biological sciences from Birla Institute of Technology and Science in India. Next, Ganesh attended Rutgers University, where he earned his PhD in biochemistry. Afterwards, he conducted postdoctoral research as a Howard Hughes Medical Institute Research Fellow at the University of California, San Diego. Ganesh served on the faculty in the Department of Biological Sciences at the National University of Singapore for over a decade before accepting a faculty position at Penn State, where he is today. His lab is recognized as a Waters World Center of Innovation in Hydrogen Deuterium Exchange Mass Spectrometry, and Ganesh was awarded the National University of Singapore Outstanding Scientist Award from the Department of Biological Sciences. He is also the former director of the Singapore National Laboratory for Mass Spectrometry and the former vice president of the Singapore Society for Mass Spectrometry. And today, Ganesh, we're excited to get to know you as a scientist, of course, but also more generally as a person. So can you tell us, what do you like to do when you're not very busy doing all of your scientific work?
I'm a musician. I'm a vocalist. So I take voice lessons, music lessons. I have been part of choirs and I sing both classical genres and light music.
I think that's amazing. How did you get into music?
I think music was the first thing I remember. I used to love harmonies and harmonizing, and my parents recognized this very early on. I think when I was 3, I would be very sensitive to the type of music that was being played on the radio in those days. Some songs I would get upset, and this was even before I was speaking. So my parents knew that they had to put me in music lessons, and so it's been wonderful learning music.
I love that. I love that you had strong opinions about music even before you could share them. Well, Ganesh, it's wonderful to hear more about your passions outside of science, but I know you're doing some amazing research as well. So how do you describe your science to people who might not be in your particular field or might be outside of science altogether?
I think of myself as being very interested in molecular switches. So if you take life processes at a molecular level, at a level that you cannot observe visually, how do these molecules interact with each other? How do they switch from one state to another? So it's like looking at very, very tiny molecular switches, kind of akin to electrical switches, but they're just so tiny. And how do they precisely get turned on and turned off is what we're very interested in.
I love it. I love the parallels that you draw between electronics and the biology. I think that helps people sort of visualize what's happening. Well, I look forward to getting into the details of your work as we go through our conversation today, but perhaps we can pause and talk a little bit about motivation. I think as a science, there is a lot to do in a day and not enough hours to do it all. So when you are struggling with something or you need a little boost of motivation or inspiration, do you have a favorite quote or a saying or something that really keeps you going, Ganesh?
My go-to quote is arise, awake, and stop not until the goal is reached. It's a saying by this saint called Swami Vivekananda, and he came to America in the 1890s and he gave a lot of speeches. And one of the quotations is this particular one. The goal is something that you have to figure out on your own. No one else is going to tell you what that goal is. And I really get inspired by this motivational quote.
Oh, absolutely. And this ethos of get up, get moving, and get it done, right?
That's right.
Well, wonderful motivational quote there. And I think as a scientist, not only do you have to motivate yourself, but you work with some remarkable people who can motivate and inspire you. So when you think about your career as a scientist, are there particular people who were role models or mentors or people who really inspired you and helped you get to where you are today?
I think it started off with my dad, who studied electrical switches, and that's why I could relate to that from a biological context. And the idea of getting into science came from my dad. And I've had some really amazing mentors from my college biology teacher, Dr. Sandhya Mitra, and my grad advisor, postdoc advisors, Betsy Kamabis at UCSD and Natalie Yan from University of Colorado, to name a couple. And just one more name is Jack Johnson. These are people that are go-tos for me. So it's very hard to name one role model, but these have been really amazing scientists who've been so generous in motivation and in being a cheerleader for your successes and to pull you up when you're struggling with something.
Definitely. I think having this network of people to help support you when the times are difficult and to kind of help shape you as a scientist during these early phases of your career is critically important. But let's talk about the beginning. You mentioned that your father was an early role model and someone who helped you discover your interest in science. How did all of this happen? How did you first start to get interested in science and realize that this is something you wanted to do?
So as a kid growing up in the 70s, I think everybody who was science-oriented or even wasn't, when you read about the moon landing, the whole Apollo space program, everybody wanted to be an astronomer. So that was the start. My dad was a physicist and he was also working in communications, in radio and satellite communications. So these were pre-internet days. I was always fascinated by how you can package information and communicate across the world, either through telephones or other media. So it was a lot of physics as a background that got me interested in it. And biology was something through school that seemed very intriguing. You felt really home when you could combine physics with biology. Biology seemed like a melting pot.
Very cool. And can you share with us, then you mentioned you had this early interest in science and maybe in college started to understand what particular areas you wanted to pursue. What then were some of the moments that you see as these pivotal moments that really helped you get to where you are today?
I think the most pivotal moment was after graduation. So during my undergrad, I was exposed to molecular genetics and how DNA decides and stores all of this information that forms the genetic basis, that molecules are really pivotal in deciding how a single cell becomes a dog or a monkey or a plant. It has all of that information. So that was initially very interesting. But after I graduated, I spent a year in a lab which was doing studies on protein folding. And for me, that's when I realized that biological molecules, the lines between biology, physics, and chemistry really blur, because you're talking about how a thread like a protein molecule is able to magically form this three-dimensional structure precisely. And that 1 year in protein folding really convinced me that proteins were where I want to be. And along the way, I also did an internship in a pharmaceutical company to watch pharmaceutical processes. And it also led me to realize that I wanted to do something more basic science rather than an application-focused science. which was interesting as well. But these more fundamental questions at the intersection of biology, physics, and chemistry seemed much more exciting. And it seemed like Aladdin's cave for the picking.
So many treasures to choose from, right?
Yeah.
Well, Ganesh, this is wonderful. And it sounds like at that point you had kind of decided that you wanted to pursue an academic career path. You decided to do your PhD. Can you talk about what brought you to Rutgers?
I was very interested in the graduate programs that were much more interdisciplinary. I was looking for molecular science programs that were not defined as one area. They were interdisciplinary. And the US graduate programs in general afford a lot of flexibility in course design and the ability to move laterally to explore in the molecular life sciences. And Rutgers had a joint program in biochemistry that had exactly that interdisciplinary feel. I did visit for open house and I really liked what I saw. It had clinical physics, chemistry, genetics, all of those being integrated in this interdisciplinary program. And so that was very attractive. And I must say that most programs that I was considering also offered the same mix of different areas of expertise where you could really learn and explore without being pigeonholed into pharmacology or biochemistry or medicinal chemistry. It was more problem-oriented, the environment. And I really liked this joint program in biochemistry at Rutgers.
Definitely. And it sounds like it was sort of ahead of its time because I think now this interdisciplinary research is becoming more and more common and you don't have to just pick one field. Oftentimes these scientists are working together from different backgrounds to solve, like you mentioned, these problem-focused questions.
Exactly. Sometimes I keep thinking, what's in a name? I'm a pharmacologist, I'm a biochemist, I'm an immunologist. But what's in a name? It's better to go beyond it. When you're studying the molecular sciences, all of these lines blur. And today it is a strength to be able to pick computational, pick different areas without being trapped in a term for what you do.
Definitely. And then when you finished your PhD in biochemistry at that Rutgers program, you went to the University of California, San Diego. Can you talk about your postdoctoral experience?
So I graduated my PhD in the year 2000. And in 1999, that was when the entire human genome was sequenced around 2000. And there was a lot of expectation, excitement, and fanfare with a lot of different expertise areas, computational, everything converging on biology at that time. So Rutgers organized in 1999 a huge structural genomics symposium, and I was really encouraged by my PI to look at all of the new developing opportunities coming in the wake of the Human Genome Project. And I had one visitor from UC San Diego, Betsy Kamavis, who was applying this chemical approach to study how proteins interact with each other. So that turned out to be a 2-week collaborative project that led to a publication, but it also convinced me that it was a perfect segue to get into postdoctoral research at UCSD. And what I was looking at different kinds of switches, these were eukaryotic protein kinases. And UCSD, the program was really very exciting. It brought in physicists, structural biologists, biochemists, enzymologists, and computational biologists to all work on this kinase. And my mentor, Dr. Susan Taylor, was excellent in bringing all these different areas of expertise to study kinases. And so I really enjoyed the multidisciplinary aspect of the postdoctoral fellowship at UCSD.
Very cool. It sounds like a fantastic experience. And then I think there's this moment where a postdoc has to decide what to do next. And that can be a stressful time and a stressful transition. So can you talk about what that transition was like for you and how you ended up at the National University of Singapore?
The UCSD program was so good, and I remember agonizing about what to do next. The UCSD model was a source of a lot of collaborations, and one of the collaborations was with Singapore Biotech Expansion and also the National University of Singapore. So they had a bilateral tie-up with UCSD, and this is just post-Human Genome Project. So as part of that, I got to meet a lot of the scientists from the National University of Singapore. There were a lot of people at UCSD that also took up positions at National University of Singapore. So I was encouraged to set up a structural mass spectrometry platform. And it was an opportunity that I couldn't turn away from, which is to start an entire new analytical approach to studying proteins. So it felt like a very exciting opportunity. I moved in 2006. It was also advantageous to be close to home. I'm from India, and Singapore is just a 4-hour flight from home. So it seemed like a great opportunity to set up this structural mass platform as well as be closer to home. So that was a big driver to moving to National University of Singapore.
And you were there for over a decade with your lab there at the National University of Singapore. What then made you decide you needed a change and you wanted to come to Penn State?
It was a very tough decision to make. I started off working on molecular switches, but around 2010, one of my mentors, Jack Johnson, was a visiting professor at Singapore, who I knew from UCSD. He strongly encouraged me to start looking at viruses. And those viruses started to get my attention. I have this little prop with me on these virus models, which I really demonstrate how viruses are not rigid rocks, but they're constantly moving around and fluctuating, which we call as virus breathing in solution. So this virus program was really successful. It's not like I stopped working on molecular switches, but I was looking at larger entities like virus particles. Around 2010, we started to work on viruses. It was an excellent program. And one of the viruses that we were studying was dengue. And I felt that there was an opportunity to expand virus research and start looking at additional viruses. And this opportunity at Penn State presented itself where they wanted to look at different kinds of viruses. So then I visited Penn State, and it was a really great environment in the infectious diseases space. And I decided that it was a great opportunity. And of course, that was before the COVID-19 pandemic, which also confirmed how valuable and important it was to look at molecular virology. So it was a challenging but important time to move to Penn State and to expand the study on virus dynamics.
Oh, definitely. And you moved to Penn State during 2020, the latter half of the year. What was that like?
I did. That was really challenging.
So it sounds like that was a difficult time, but things are going well in your lab at Penn State today. And I'd love to talk more about some of the research that you're doing there. So Ganesh, is there a particular project that you are working on right now that you are just the most excited about and want to share with us today?
I have two research programs, one on signaling proteins. Those are the molecular switches that I've continued from my San Diego days. The other is on viruses and how viruses change their shapes depending on whether they're getting ready to infect a host or not. So viruses don't have any sensory organs. So one of the questions we were most interested in is they don't have eyes or ears. How does a virus figure out whether it has entered a host environment or it's somewhere on the outside? For instance, if you have a virus that causes gastrointestinal symptoms, and it usually exists in contaminated water, which is a very different environment from when it's inside a host's gut. Now, how does a virus figure out whether it's in contaminated water on the outside or if it's inside a host's gut? How does it change its shapes accordingly? So it turns out that every virus uses its coat as a thermodynamic sensor. So the environment in contaminated water outside is going to be a colder temperature, but when it enters the gut of a human host, it's going to be a warm-blooded 37 degrees. It's going to be a very different pH and salt concentration on the outside versus the inside. So this virus is able to factor in the altered environment to decide it's a good place to start replicating, to shed its nucleic acid and start to make multiple copies of itself. And every virus does this because every virus is traversing different environments in its life cycle. And one of the very interesting areas we're studying is a plant virus. And this plant virus, we have captured the particle in the act of releasing its nucleic acid inside the host cell. So it's a little volcano that forms. It's like a miniature seed. And the RNA, which is its nucleic acid genome, is able to trickle out kind of like a soda can. The RNA only comes out of one end of the particle. It doesn't come out in all directions. So there's a structure and an orientation to how the soda can is built. And we are using analytical chemistry probes to understand how are these virus soda cans made, how are they assembled, and how is that little notch on one end designed so that the RNA trickles only from one end to the outside. So I'm very excited by understanding this question of how viruses are assembled. All viruses contain nucleic acids or RNA as their genomic material. And it's intriguing how precise this process is. There's no chance that any of the host cell's RNA is packaged into viruses. It's always the virus packaging its own RNA. And we're getting close to understanding what those first principles are for how RNA self-packages itself. So that's what keeps me up at night. So that's what I'm most excited about right now.
Well, Ganesh, this is absolutely fascinating. You have us on the edges of our seats here. It also has important implications downstream for public health, being able to just understand the basic processes and transformations that these viruses undergo. So very excited about the work that you're doing, but I know it's not all fantastic victories and exciting science and exciting days in the lab. You have frustrations, you have challenges, you have struggles, and I think these can be tough to get through as a scientist. So when you feel like everything is going wrong, how do you get through these major challenges or failures? And do you have a particular example you could tell us about?
There's always self-doubt when you're starting something new. One example I had was before writing the first paper, my first publication as a graduate student, there was always this doubt of what if? What if you're not going to be able to get transformants that you need, which is a critical keystone for your project? For you to be able to write something, you need that one experiment to really succeed. And I remember trying it on a Saturday, and my PI came over and said, "Did it work?" And I said, "No." So there was that look about, we have nothing to write about because that was the really pivotal experiment. But I don't know what it was. This sort of dogged optimism that made me want to try it again, and finishing that experiment at 2 a.m. and getting the results on Sunday. And when the PI walked in on Monday saying, "I don't know what we're going to do," and I said, "No, it worked." So that self-doubt, to be able to park that and not overthink what if it didn't work, I think that's something that is a very inspiring feature of American entrepreneurship, whether it was the moon landing or whether it was the first flight across the Atlantic. There was never a thought about what if this isn't going to work. Not that it was there, but it was always parked aside when people tried it, whether it's the first person who climbed Mount Everest. And there's always these instances and biographies. So I think it's been self-doubt. And again, when I moved into the virus field, I was thinking, I haven't worked on viruses before. I've only been working on signaling proteins and switches. Isn't this a totally new area? How am I going to be able to figure this out? It looks very complicated. But I think it's always been about parking your self-doubt, acknowledging it, but not letting it get in the way.
Definitely. I love these stories. I think you're absolutely right. You always have that nagging voice in your head, which can be constructive sometimes, but you can't let that stop you from moving forward in your science. And I have to ask, did you ever figure out why that one experiment on that Saturday didn't work the first time?
No, I couldn't, because it's an experiment called a transformation. There's many variables that can be wrong. And one of the things is I was trying to do 7 at the same time. Instead of doing it linearly, we were on a time crunch. And I just think it was the self-doubt. It's not a very quantitative answer, I know, but I couldn't figure out what that parameter was.
I think luck was on your side the second time, which is often the case. I think in science, you have to factor luck into the equation.
Yeah, but I kept getting from one of my friends who's always luck favors the prepared. I don't know, that was just sort of dint into my head.
Well, I think that's wonderful. And it sounds like this particular story in both cases had a happy ending. You were able to work through that self-doubt. And I think it's so important to celebrate the wins in science because so much work goes into them. So do you have a favorite success story, Ganesh, that you would like to tell us about, whether it was one of these huge wins or just a small but really meaningful one?
The most exciting part was this recent story on the protein kinase A, how protein kinase A resets itself after stressful situations. I think this paper that was just published last month was very satisfying because it was a 3 to 4 decade elusive search for what the features of this stress reset molecule look like, and solving it and figuring out that it's more like a fuzzball rather than a rigid rock was really, really satisfying. And to be acknowledged by stalwarts in the field in JACS, which is the Journal of American Chemical Society, was very gratifying because a lot of the major breakthroughs in the molecular sciences have been in JACS. And so that's been a first and a very satisfying feeling.
Definitely. And I think when you have a major breakthrough, oftentimes it can be met with skepticism in the field. Did you encounter this, or were people pretty on board with the results that you published?
They were on board with it partly because it had a 30-year-old history of being very difficult to lock in through static viewpoints. You had to have it was this fuzzball that was constantly moving around. So it sort of connected all the dots, and it needed somebody to connect those dots. And because it was a 30-year-old problem, there wasn't much of skepticism for it. It raised the opportunity of looking at reducing some stress-induced effects on carbohydrate metabolism, the potential for what the impact of weight loss drugs and glucagon-like peptides might be on how the system resets. So there's a lot of opportunity to look at modern-day stressful living and modern-day challenges with obesity and other related diseases.
Oh, very cool. Well, Ganesh, it sounds like this explained a lot of the challenges that people had been having in previous decades, as well as opened a lot of doors for new questions and new research. So congratulations on this meaningful success in the field.
Thank you.
How did you celebrate?
We had a lab party, and it was a dinner party for all of the lab students and graduate students.
What kind of food did you get?
We had 2 parties, one with Indian food and one with American diner food at Penn State.
Fantastic. Well, congratulations again to you and everybody involved in this project over the years. And I know these successes can get you really geared up to work even harder in the lab, but we try to encourage people to also give their mind some space to take a break and to read if they can. So we always are looking for recommendations from our guests on the show to share books that they've enjoyed, whether they're related to science or not. So Ganesh, do you have a favorite or even just one that you've read recently that you'd like to recommend for us today?
The book that I really liked that I've read very recently is called The Empath's Survival Guide: Life Strategies for Sensitive People by Judith Orloff. I have understood that I am a sensitive individual, and I've been always very intrigued by this character in Star Trek called the empath, the one that is able to feel another person's situation. And it comes with a lot of responsibility and intuition and a lot of strengths, but it can also be overwhelming in terms of how much information you get. So I benefited enormously by reading this book because it sort of connected me with the neuroscience of empathic behavior, what makes a better mentor, how do you be a better colleague, and how do you listen, and how do you make sure that you're not taking on more than you should? How do you use nature and alone time to regenerate and refresh? I think it's been a wonderful book. The other book that I've heard a lot about, but I'm very interested in finishing fully, I haven't yet completed the book. It's called Flow, The Psychology of Optimal Experience by Mihai Csikszentmihalyi. I hope I'm not mispronouncing, but it's about the flow state. And I'm very interested in how all individuals can access the flow state. So these are two areas that I'm very interested in.
Fantastic. These are excellent recommendations, and I believe you are the first to recommend each of these books. So we will add them to our website for our listeners to find there, The Empath Survival Guide and Flow. And we talked about some different aspects of your career. And in talking about even just your academic training, we highlighted all of the different places that your science has taken you, Ganesh. You've been around the world. And I think not just for your academic training, also for conferences to work with colleagues. So can you share with us, what is one of your favorite places that you've been to for science, and what made that place so memorable for you?
One of the places that I really enjoyed a lot was Copenhagen, Denmark. I was invited to be a PhD student examiner for a student who was receiving his PhD, and I was invited as an external examiner to evaluate the dissertation. And it was an eye-opener because it was a much more prolonged ceremony. You got to meet the student candidate's family, and you went and saw where they worked. It was a great experience. And I also got to explore Copenhagen, and I went to Hamlet's Castle. It was just a very magical journey as part of this 3-day examination of a PhD. I learned a lot about the culture and history there. It was really nice. It was very well laid out. And if I may, the other place that I enjoyed was a month's stay that I had at the Indian Institute of Science in Bangalore, where I could just be myself and interview a lot of different scientists and learn about their work. And it was this really bucolic campus, and I enjoyed myself a lot. There were no administrative pressures. It was a sabbatical for a month, and I really enjoyed myself. So you never get those opportunities, but when they do come, they're very satisfying.
Definitely. So if our listeners are able to make it to either Copenhagen or Bangalore in the future, what are places that they must see or sites that they have to visit when they're there?
Copenhagen would be Hamlet's Castle, which is a little, maybe an hour's drive away, but it's really nice to watch the castle and all of the royal items there on display, and just to look at the narrow straits that the kings used to collect the tolls for traffic going inland into Russia, Finland, the Baltics. So it was a very strategic location that made the Danish kings really wealthy. It was a fascinating place geographically to observe. And in Bangalore, it would be the IISc, the Indian Institute of Science campus. One of my aspects that I really liked was the architecture and the greenery, as well as the government administrative buildings there. And wonderful weather, really great space to have scientific discussions, and it was very educational. One of my favorite conference locations is Taos, New Mexico. It's an absolutely beautiful location in the Southern Rockies. Taos has a beautiful pueblo, and there's also these beautiful overlook points where you look at the Rio Grande River, and it's just absolutely magnificent as a place to hold conferences and interact with other scientists amidst beautiful nature.
Well, this sounds like a breathtaking conference destination. And all of these places that you mentioned sound absolutely amazing. And Ganesh, I'm so glad you were able to visit them. It's been wonderful to hear about this scientific travel, but I think you also get to work with so many cool people from around the world as a scientist. And I think the people that you meet often go against the stereotypes that people may have of scientists being very serious and very critical and always working in the lab in their white coat all the time. And I think there are so many instances where this is not the case in science. So, Ganesh, do you have examples of quirky lab traditions or just funny or fond memories that you shared with colleagues that may go against the stereotypes that people may still have about scientists?
Absolutely. So in Rutgers, we had a ritual where every day at 3 p.m., you had to go drink coffee, and it was a group activity with a bunch of students. You had to drop what you're doing. And sometimes it used to be bigger forum where everybody on the floor gathered together. But every 3 p.m., you had to stop what you're doing. And it was really great because it was camaraderie. And sometimes it would be talking about American Idol the previous night or where people were going. But it sort of added a really nice human touch to scientists and what we do, and just to lighten the mood. And in terms of a funny story, when I was just starting off as a new PI in Singapore, we went for lab lunch, and I had a student who was mistaken to be the PI, and the check for the meal was given to my grad student. And that was the day that I just laughed and laughed because and the student was so upset. He was heavily built, and overnight he stopped eating fast food and went on this crash diet because of that incident where he was mistaken to be the PI. So I still give him a hard time after 20 years. I still affectionately call him uncle because he looked like my uncle enough to fool the waiter at the restaurant to think that he was the PI somehow. But he transformed into this nephew. So we still joke about how that was his inspiration to eat healthy.
I love this story. It sounds like a life-changing moment when he lost the lunch lottery there. Well, I think this highlights just the personalities in science, both of your examples here, and just the fact that it is a team effort. You are working with these people kind of towards the same mission to solve these big problems in the lab. And I think it's helpful to have people who have this sense of humor and are there for you along the way, because you're dealing with limitations in a lot of cases, whether it's funding, staff, technology, time. So if we took away the barriers that normally stop you, Ganesh, what is the one research question that you would want to answer most?
It would be, how does RNA package itself? I know it's so specific, but it's how do molecules self-assemble, to put it in broader terms? How do they have the principles to self-assemble? I find that fascinating.
Absolutely. Do you have hypotheses of what might be happening or driving this?
The analogy I can give is there's a special type of glass art where you start off with the nucleation on one end, and then the rest of the glass crystallizes and condenses on itself. And it is never meant to be a perfect geometry, but there's always these imperfections when you look at how molecules self-assemble. So it might seem like some yarn, but somehow if you provided the right nucleation, like if you want to tie up yarn and organize it into bundles, you usually use some kind of nucleating center point around which you wrap the loose threads in order to give you a ball of yarn. I think something similar happens in all of these molecules which learn to self-assemble. And the key is going to be how to identify what is that nucleating element that allows you to form these organized clusters.
Oh, fascinating. Well, Ganesh, this is a cool mystery for me and our listeners to contemplate this afternoon. So thank you for sharing it with all of us today. And before we let you go today, Ganesh, we would love to talk about advice. And to start off with, I'd love for you to share the best piece of advice that you were ever given so our listeners can benefit as well.
I think my best advice is to acknowledge self-doubt, but then always compartmentalize it so that you can park it aside. You have to sort of acknowledge the voice in your head, which says, oh, it may not happen. And then give yourself moments where you're not thinking about what if, and always look at the positive side of things. I think ultimately any solution for any scientist is going to be to look deep within and trust your intuition and not let self-doubt take over.
Well, Ganesh, that is a powerful message for our listeners out there. Is there any other last piece of advice or last note of inspiration that you'd like to leave everybody with at the end of our conversation today?
I think resilience is very rewarding. Everybody has the same amount of time, but looking at role models, looking at biographies of people who have made it is always the way to go. And also learning is a lifelong process and mentoring is lifelong. So you have to keep looking out for mentors who are your cheerleaders, who understand you and who will celebrate your successes and pull you out when things aren't going your way. So always keep looking for and cherishing your lifelong mentors. It doesn't end in school or the university, but learning is lifelong and finding good mentors is a lifelong endeavor.
Definitely, listeners, keep learning and keep building this community of mentors and role models and people who you're mentoring as well. I think you can learn so much from both sides. So that is a wonderful message for us to end with today, Ganesh. If our listeners would like to learn more about you and the work that you're doing, how can they learn more or get in touch with you?
They can reach me via email. My research group has a website as well as we have a presence on X and LinkedIn too. So all of these are for
Excellent. Well, listeners, definitely take some time to learn more about the remarkable work that Ganesh and his team are doing. Get connected with him on social media. And Ganesh, it's been such a pleasure to chat with you today. Thank you so much for your time.
Likewise, and I love what you do.
Oh, thank you. I'm very excited about the research that you're doing. And listeners, we're thrilled to have you here with us today. And we hope you can join us next time for another episode of People Behind the Science.